Welcome back to CRISPR Corner. Last episode, I gave you some rules to follow, or maybe break, when designing CRISPR guide RNAs. In this episode, I'm going to give you a demo of one of the online tools that I use to design guide RNAs. First, navigate here. For this demo, we'll be using the Unlocked CRISPR Design tool, which you can get to by clicking on the icon. To unlock all of the features in the tool, create a free account. First, you'll need to input the target genome. This is the species that you'll be working in. Next, you can design either based on a specific gene using the gene symbol, gene ID, or alias, or you can copy and paste a sequence into the tool using the input sequence option. If using a specific gene, tell the tool if it is a protein coding gene, microRNA, or long non-coding RNA. Then type the name of the gene into the box and select the correct line from the drop-down. Next, you'll need to select the transcripts that you want to target. If you're going for a true knockout, leave them all selected. If you're using Cas9 to create a double-stranded break, you'll leave that selected too. And I suggest leaving the guide RNA mismatches at 3 base pairs or more. This goes back to our design rules and helps reduce potential off-targets. Once you've got everything into the tool, hit submit. After a bit of thinking, you'll be taken to your results page with numerous guide designs to knock out your gene of interest. From here, you can peruse your designs on this page, but it can be a lot of data. So I usually like to use the Download All button to download a spreadsheet of my designs for easier sorting. Your downloaded file will look something like this. I did play around with some of the column widths to make some things easier to read. If you're working in a program other than Microsoft Excel, these next steps may look a little different. First, select the entire spreadsheet and unmerge. Then, I like to get the columns I use most next to one another. Those columns are efficiency, specificity, number of transcripts hit, and start coordinate. Not sure why those are the ones I use? Check out my previous episode on guide RNA design. You can find all CRISPR Corner videos by searching CRISPR Corner on YouTube. Alright, from here, I'm going to do a custom sort of my guide designs. Select the full table of guide designs, including headers, and click on Sort and Filter and Custom Sort. And make sure that My Data Has Headers box is clicked. Then sort Specificity Z to A, then Efficiency Z to A. Based on how it downloads, you're likely to get this warning. Choose to sort anything that looks like a number as a number and click OK. From here, I'm going to look at my highest specificity guides. For this gene, we've got a lot of great options with a specificity score of 100, meaning all potential off-target sites have more than 3 base pair mismatches with the guide sequence. I then look for high specificity guides with an efficiency score greater than 40. That leaves us with these designs. In this case, my gene only has one known transcript. If your gene has multiple transcripts, you'd also want to ensure your top-ranked guides hit every transcript. From here, I would pick at least 3 guides to test in my experimental system to generate the knockout. The last thing I'd look at when deciding which to test would be the start coordinates to ensure I don't have overlapping guides. For this set, we have 2 guides that are 1 base pair away from one another. For these, I'd choose the one with the higher efficiency score. And there you have it. If I was looking to knock out this gene, I'd start with any of these highlighted guides and feel good about my decision. The gene in this walkthrough was well behaved for CRISPR guide design. Don't forget that other genes might require a bit of rule breaking. If you're ever faced with a particularly tricky design, reach out to our Millipore Sigma team. We can provide advice on guide design and the rest of your CRISPR workflow. In a future episode of CRISPR Corner, I'll show you how you can use this same design tool to search for pre-designed databases for guides for gene knockout, gene activation using CRISPR-A, and gene inhibition using CRISPR-I. Until then, happy CRISPRing!